in the vast and silent expanses of the world's oceans, where the pressure of the deep water creates a world of stillness and the sunlight fades into a permanent twilight, there exists a biological anomaly that challenges every fundamental law of life and death we have ever established. Scientifically classified as Turritopsis dohrnii, but more famously known across the globe as the Immortal Jellyfish, this tiny hydrozoan is a master of survival that hides its revolutionary secret within a body that is almost entirely composed of water. When we look at this creature, which measures no more than 4 or 5 millimeters in diameter, it is difficult to imagine that such a fragile and translucent bell could hold the key to biological immortality. The journey to understanding this creature began in the late 19th century, specifically in the year 1883, when a German zoologist named August Weismann first described the species after observing it in the warm currents of the Mediterranean Sea. At that time, the scientific community viewed it as just another small inhabitant of the complex marine ecosystem, a simple organism with a standard life cycle that would eventually lead to senescence and death. For over a century, the Turritopsis dohrnii remained in the shadows of academic archives, categorized but not truly understood. Why it quietly drifted the oceans from the Caribbean to the coasts of Japan. It was not until the late 1980s, during a routine laboratory observation in Italy, that the veil of mystery began to lift. Researchers led by Fernando Boero and Giorgio Bavestrello were keeping a colony of these Medusae for study, when they noticed something that defied all biological logic. When the environment became unfavorable, or when the jellyfish faced physical trauma or starvation, they did not perish as any other animal would. Instead, they began to undergo a process that looked like time itself was being folded back upon itself. The adult Medusa, with its pulsing bell and delicate tentacles, would slowly begin to shrink, its tissues would reabsorb, and it would transform back into a cyst-like state before eventually returning to its very first life stage, the colonial polyp. This was not mere regeneration, it was a total cellular reset, equivalent to a butterfly turning back into a caterpillar or a fully grown tree shrinking back into a tiny seed. This phenomenon, known as ontogeny reversal, shocked the world of biogerontology. It suggested that the linear progression of time from birth to death was not a universal mandate for all complex life. The discovery forced scientists to rethink the definition of an individual, because this jellyfish could theoretically cycle between its juvenile and adult stages indefinitely, avoiding the biological decay that characterizes the end of life for almost every other multicellular organism on Earth. As word of this immortal creature spread, it became a focal point for geneticists and molecular biologists who sought to decode the specific signaling pathways that allow a specialized cell to lose its identity and regain the potential to become any other type of cell. This process, which we now identify as transdifferentiation, is the engine behind the jellyfish's ability to cheat death. Through the decades since its discovery, the Turritopsis dohrnii has become a symbol of the hidden possibilities within nature, a reminder that the deep sea still harbors secrets that could redefine our understanding of medicine, aging, and the very nature of existence itself. While it drifts through the dark, cold currents of the global oceans, it carries within its microscopic frame a blueprint for a life without a biological end. A silent witness to the fact that in the realm of science, the most extraordinary truths are often found in the smallest and most unassuming forms of life. And as we continue to peer into its transparent structure, we find ourselves looking into a mirror that reflects our own quest for longevity and the eternal mystery of the cycle of life. The complex life cycle of hydrozoans, the diverse class of cnidarians to which the immortal jellyfish belongs, represents a remarkable adaptation to the marine environment, standardly characterized by an alternation of generations. This intricate process begins far from the pulsing, free-swimming form commonly associated with the word jellyfish. The standard cycle initiates with sexual reproduction, typically involving adult medusae releasing gametes, sperm and eggs, into the water column, where external fertilization occurs, forming a zygote, and this zygote quickly develops into the first key life stage of the organism, known as the planula. The planula is a microscopic, flattened, oval-shaped larva covered in tiny, hair-like projections called cilia, which allow it to swim or rather drift through the water column. Seeking a suitable substrate to begin the next phase of its life, this larval stage is purely dynamic, designed for dispersal and selection. As the planula uses sensory capabilities to locate an ideal environment, usually a hard surface on the seabed, away from the currents, safe from heavy predation. And during this time, the planula does not feed, relying entirely on internal energy reserves inherited from the egg, its sole purpose being to find a permanent home. Once a suitable location is found, the planula attaches itself firmly to the substrate and begins a profound metamorphosis, transforming into the next fundamental stage, the polyp. The polyp stage represents a sessile, anchored existence, and often forms the longest-lasting phase in the standard hydrozoan life cycle. A single polyp has a relatively simple vase-like body structure, affixed to the seabed by a base or root-like structure known as a stolon, and extending upward with a central column terminating in a mouth surrounded by tentacles. These tentacles are equipped with specialized stinging cells, or nematocysts, designed to capture microscopic plankton or other small organic particles from the surrounding water currents, directly channeling food into the gastrovascular cavity, the central digestive region of the polyp. Unlike the Medusa, the polyp is designed entirely for feeding and growth, efficiently processing nutrients from the abundant microscopic life of the deep seabed. But the life of a polyp is rarely solitary. Through asexual reproduction, specifically the process of budding, a single anchored polyp can replicate itself, giving rise to clones that remain interconnected by the stolon network, eventually forming a complex, branching colonial structure. This colonial polyp network can grow quite large, spreading across available surfaces on the ocean floor, sharing a common digestive system. And in this unified structure, individual polyps often specialize for different functions. Some, the gastrozooids, are dedicated solely to feeding, gathering nutrition for the entire colonial entity, while others, the gonozoids, are specialized for a different kind of asexual reproduction, a process called strobilation or budding of medusae, which eventually releases the next generation back into the water column. The polyp phase, with its anchored, feeding-focused and asexually reproductive nature, provides the foundational resilience and nutritional support necessary for the hydrozoan life cycle to proceed, allowing the organism to colonize territory, efficiently gather resources, and multiply its numbers long before the delicate, pulsating Medusa stage begins its brief journey through the vast pelagic ocean. This complex and effective mechanism, from drifting planula to anchored polyp colony, ensures the persistence and distribution of the hydrozoan species in the often harsh and competitive environment of the global oceans, providing the vital baseline for understanding the unique reversal that Turritopsis Dohrnii will perform. The morphological transition from a sessile anchored polyp colony to the motile, free-swimming Medusa stage represents an extraordinary feat of biological engineering that facilitates the dispersal and sexual reproduction of the species. This process begins within the specialized reproductive units of the colony called gonozoids, which operate as concentrated centers of asexual proliferation. Inside these structures, the organism initiates a complex series of cellular reorganizations to bud off miniature medusae. These tiny, immature forms initially lack the full complexity of the adult but already possess the fundamental blueprint of radial symmetry. As the budding Medusa matures while still attached to the parent colony, it develops a primary bell structure known as the umbrella, which is composed largely of a non-living jelly-like substance called mesogalea. This mesogalea provides the necessary buoyancy and structural integrity for the organism to navigate the fluid environment of the open ocean. It consists of a matrix of proteins and water, providing a flexible yet resilient frame that can withstand the pressures of the deep. The edge of the bell becomes equipped with a muscular membrane called the velum, a unique characteristic of hydrozoans that narrows the opening of the bell to increase the velocity of the expelled water, thereby enhancing the efficiency of its jet propulsion system. This allows the Medusa to move with surprising speed and agility despite its seemingly fragile appearance. Simultaneously, the internal anatomy of the budding Medusa organizes into a series of radial canals that extend from the central stomach to the circular canal at the bell's margin, ensuring the distribution of nutrients to all tissues. These canals act as a primitive circulatory system, moving digested organic matter throughout the organism's body. The development of the nervous system is equally specialized, transitioning into a decentralized nerve net that includes specialized sensory clusters at the margin of the umbrella. These clusters, including light-sensitive ocelli and gravity-sensing statocysts, allow the Medusa to orient itself within the three-dimensional space of the water column. The ocelli can detect changes in light intensity, guiding the jellyfish toward the surface or away from predators, while the statocysts provide a constant sense of up and down. Once the maturation process reaches a critical point, the young Medusa detaches from the gonozoid and enters the pelagic realm as an independent predator. As the Medusa grows in size and complexity, it develops gonadal tissues located along its radial canals, which are responsible for the production of gametes through the process of meiosis. The onset of sexual maturity is marked by the capacity to produce either eggs or sperm, depending on the sex of the individual, the timing of the sex of the individual. The timing of gamete release or spawning is a highly regulated event often triggered by precise environmental stimuli, such as the subtle shift in light intensity at dawn or dusk or specific fluctuations in the ambient water temperature. When these cues are detected, the Medusae release their gametes into the surrounding seawater, where fertilization occurs externally. This chemical interaction between sperm and egg is guided by species-specific attractants, ensuring that the zygote is formed only between compatible gametes. The resulting zygote immediately commences rapid mitotic divisions, passing through a hollow blastula stage before transforming into a ciliated, free-swimming Planula larva. This larva will eventually settle on a suitable surface to establish a new polyp colony, thereby completing the traditional reproductive loop of the hydrozoan life cycle. The adult Medusa phase, while biologically focused on the dispersal of genetic material, is also a period of intense metabolic activity. As the organism must constantly capture prey to sustain its energy-intensive swimming and reproductive functions, the tentacles, lined with cnidocytes or stinging cells, serve as the primary tool for subduing microscopic prey, while the rhythmic contractions of the bell maintain the flow of water necessary for both movement and respiration. This phase of the life cycle is designed for expansion and genetic mixing, providing the species with the ability to colonize distant regions of the ocean. Yet, it is also the stage where most hydrozoans eventually reach the end of their biological lifespan, succumbing to the inevitable process of senescence after their reproductive duties are fulfilled. In the specific case of the immortal jellyfish, this stage is not an end, but a potential turning point, where the standard rules of biology will be rewritten by the unique cellular mechanisms that allow for the reversal of this entire developmental sequence. Cellular transdifferentiation, which represents one of the most profound and enigmatic mysteries within the modern biological sciences, involves a specialized somatic cell losing its terminal phenotype to transform directly into another type of functional cell, in the context of the ontogenic reversal seen in Turritopsis Dohrnii. This process is the fundamental engine that powers its biological immortality, challenging the long-held central dogmas of developmental biology. Typically, in complex multicellular organisms, once a cell undergoes differentiation and becomes specialized as a muscle cell, a nerve cell, or a skin cell, its developmental fate is considered sealed and irreversible. However, the cells of the immortal jellyfish possess an extraordinary level of plasticity that allows them to bypass these seemingly rigid genetic constraints. During the transition from the floating Medusa stage back to the anchored Polyp stage, the organism initiates a radical restructuring of its entire cellular architecture. The specialized cells of the exumbrella, such as the striated muscle cells that facilitate the rhythmic pulsing of the bell, along with the epithelial cells of the digestive system, begin to lose their structural identity. This is not a simple case of cellular death or decay, but rather a sophisticated and highly regulated reprogramming of the cell's internal genetic software. As the physical bell of the jellyfish begins to shrink and its tentacles are slowly reabsorbed, the internal organelles within the cells are reorganized and the cytoskeleton is dismantled and rebuilt. The process of transdifferentiation is distinct from the differentiation, because it often involves the direct conversion of one mature cell type into another without necessarily returning to a completely undifferentiated, pluripotent stem cell state, though a brief, unstable intermediary phase may occur. This means that the very same cells that once functioned to propel the jellyfish through the water column are systematically repurposed to become part of the new polyp colony's digestive or reproductive structures. The molecular mechanisms governing this transformation involve a massive shift in the transcriptional landscape of the cell, where genes associated with the previous identity are silenced and a new suite of genes associated with the polyp stage are activated. Specific signaling pathways, such as the ancient and highly conserved Wnt signaling pathway, are thought to play a critical role in directing this cellular metamorphosis, orchestrating the complex dance of proteins and enzymes that allow the tissue to effectively melt and reform. The extracellular matrix also undergoes a complete renovation, providing a new skull for the transforming cells to settle into their new roles. This cellular fluidity ensures that the organism does not merely heal from injury, but fundamentally resets its entire biological clock, as the old, potentially damaged specialized cells are replaced by fresh, functional versions. Through this direct conversion, this ability to recycle cellular material and redefine functional roles on a massive scale is what allows the Turritopsis Dohrnii to escape the inevitable decline of senescence, providing a biological blueprint that researchers in regenerative medicine and biogerontology study with intense fascination, hoping to one day unlock similar potentials for tissue repair and longevity in other species. The sheer scale of this transformation, happening silently within the microscopic layers of the jellyfish's transparent body, is a testament to the incredible versatility of life at its most basic level, where the boundaries between different cell types are not walls but fluid gateways, and where the end of one life stage is simply the raw material for the beginning of another. In the quiet darkness of the ocean, this process of transdifferentiation continues to rewrite the rules of existence, turning the finality of aging into a temporary state that can be navigated and overcome. As we peer deeper into the molecular signals and genetic switches that control this miracle, we find ourselves at the edge of a new understanding of what it means for a cell and an organism to be truly resilient in the face of time. The concept of biological immortality, often referred to in the specialized realms of academic and evolutionary biology as negligible senescence, represents a truly extraordinary state in which the statistical probability of mortality from physiological aging remains stable or even decreases as the organism continues to exist through time, a phenomenon that stands in direct and stark contrast to the chronological aging process observed in the overwhelming majority of complex multicellular life forms inhabiting our planet. for most known species, ranging from the smallest insects to the most complex mammals. Aging is viewed as an inescapable and unidirectional progression, a steady journey characterized by the gradual and cumulative accumulation of oxidative cellular damage, the progressive shortening of protective telomeric sequences at the ends of chromosomes, and the inevitable decline of systemic physiological functions. However, the species Turritopsis dohrnii stands as a radical and singular exception to these seemingly universal biological norms. By actively and systematically rejecting the standard trajectory of senescent decay through its unique and highly specialized ability to undergo complete ontogeny reversal, this remarkable hydrozoan demonstrates to the scientific world that the biological clock is not a rigid, one-way mechanism, but rather a highly programmable and flexible system that can be periodically reset to its primordial juvenile state. At the deepest molecular level, this rejection of senescence involves the sophisticated maintenance of genomic integrity and the active prevention of the cellular state known as the Hayflick limit, a biological boundary which typically dictates the maximum number of times a somatic cell can undergo division before it enters a state of permanent growth arrest or programmed cell death. While the cells of most organisms eventually lose their capacity to repair their double-stranded DNA breaks efficiently, leading to the functional and structural degradation that we perceive as the outward signs of aging, the individual cells of the immortal jellyfish appear to possess a specialized and highly active suite of protective enzymes, antioxidant proteins, and complex DNA repair pathways that remain functional and vigilant throughout its multiple life cycles. By periodically reverting from the sexually mature Medusa form back to the juvenile, anchored polyp stage, the organism effectively clears the accumulated metabolic waste products, damaged proteins, and detrimental epigenetic markers that were acquired during its free-swimming adult phase, essentially purging the biological signatures of time from its entire system. This does not imply that the individual organism is invincible to all external threats, as it remains susceptible to the dangers of predation, disease, and catastrophic shifts in its local marine environment, but it does mean that there is no internally mandated, genetically encoded expiration date within its specific genome. The rigorous study of this phenomenon profoundly challenges the traditional evolutionary theories of aging, such as the disposable soma theory, which posits that organisms must strategically allocate their limited energy resources between the demands of reproductive and the maintenance of their non-reproductive tissues, resulting in an inevitable decline in repair as the organism matures. Turritopsis dohrnii effectively disrupts this evolutionary balance by investing in a total and systemic overhaul of its body plan, allowing it to bypass the biological cost of aging through a continuous process of recycling and renewal. This suggests a level of evolutionary efficiency that allows the species to persist in a state of perpetual potential, never reaching a terminal stage of existence. The process of ontogenic reversal, which serves as the defining and most scientifically significant characteristic of the species Turritopsis dohrnii, represents a profound and radical departure from the standard biological progression observed in almost all other multicellular organisms on Earth. In this extraordinary phase, the sexually mature Medusa, having already achieved its fully developed adult form and perhaps even engaged in sexual reproduction, begins to undergo a structural and cellular collapse that effectively rewinds its biological history and resets its life cycle. This unique event is typically triggered by extreme and life-threatening environmental stressors, such as a sudden and drastic drop in water temperature, a significant decrease in salinity levels, physical trauma to the delicate umbrella or tentacles, or the prolonged onset of starvation in nutrient-poor currents. When the Medusa perceives these critical conditions, it does not simply succumb to the inevitable process of mortality as other hydrozoans would, but instead initiates a highly programmed and genetically encoded response designed to preserve its genetic material through a total systemic reset. The physical manifestation of this reversal begins with the gradual contraction and degradation of the umbrella. The rhythmic and elegant pulsing that once propelled the jellyfish through the water column becomes increasingly irregular and then ceases entirely, as the muscular tissues of the velum lose their vital tension and the bell begins to shrink inward upon itself. Simultaneously, the long and fragile tentacles, which were once highly specialized tools for predation and defense, are slowly and systematically reabsorbed into the main body mass of the Medusa, their complex structures breaking down at the microscopic level as the stinging cells or cnidocytes are deactivated and dismantled. This dramatic reduction in morphological complexity results in a state known as the cyst-like stage, a dense, amorphous, and undifferentiated mass of biological tissue that eventually loses its buoyancy and settles onto the dark ocean floor or attaches itself firmly to a stable substrate such as a rock or a discarded shell. Inside this tiny, protective, and sphere-like structure, the process of transdifferentiation reaches its peak metabolic efficiency, working to convert the specialized tissues of the adult Medusa into the foundational and pluripotent-like cells required to build a completely new polyp colony. The internal organs, including the central stomach and the intricate network of radial canals, are reorganized into a primitive gastrovascular cavity, and the once active reproductive tissues are entirely dismantled. As the organism shifts its entire energy budget from sexual proliferation to a survival-oriented asexual regeneration, this transition from a highly active, free-swimming, and high-energy life form to a sessile, low-metabolism cyst is a masterwork of evolutionary ingenuity, providing a biological refuge in which the individual can wait out unfavorable environmental conditions before emerging in its juvenile form. After a period of internal restructuring, the cyst eventually begins to extend new stolons, the root-like structures that anchor it to the seabed, and from these stolons new polyps begin to bud and grow, effectively starting the life cycle over from the very beginning. This is not merely a birth, nor is it a simple recovery from physical injury, but the fundamental rebirth of the exact same genetic individual in a younger and more resilient morphological state by collapsing its complex adult structure and rebuilding it from the basic cellular level. Turritopsis dohrnii successfully navigates the boundary between life and death, proving that biological complexity can be deconstructed and reconstructed as a primary survival strategy. The silent and invisible transformation occurring within the dark, cold depths of the ocean, beneath the crushing pressure and eternal shadows, is a masterpiece of cellular engineering where the adult form is treated as a temporary vessel to ensure the persistence of the biological essence. Through this ontogenic reversal, the jellyfish avoids the terminal endpoint of the standard hydrozoan life cycle, trading the mobility of the Medusa for the durability and longevity of the polyp. As the new colony begins to expand across the seabeds, it carries with it the exact same genetic blueprint as the Medusa that preceded it, but with a completely refreshed cellular clock, ready to face the challenges of the marine environment once again. This process remains one of the most intensely studied phenomena in the field of biogerontology, as it holds the potential secret to a controlled and systemic reversal of the aging process, a feat that remains far beyond the reach of almost all other life forms in the known universe. The sheer elegance and mystery of this reversal, from a pulsing creature of the open sea to a quiet, anchored inhabitant of the ocean floor, highlights the fluid and non-linear nature of identity and time in the life of the immortal jellyfish. The extraordinary phenomenon of cell plasticity, which serves as the primary biological engine behind the life-extending capabilities of Turritopsis dohrnii, represents a profound departure from the rigid cellular hierarchies that govern the development and aging of most complex multicellular organisms. In the standard biological model, as a cell matures and specializes into a specific role, such as a muscle fiber, a digestive cell, or a stinging cnidocyte, It undergoes a process of terminal differentiation that is typically viewed as a permanent and irreversible loss of developmental potential. This specialization is enforced by a complex layer of epigenetic modifications that lock the cell's genetic program into a specific functional state, effectively silencing the genes associated with earlier or alternative stages of development. However, the cells of the immortal jellyfish possess an inherent and remarkable capacity to dismantle these epigenetic barriers, allowing them to regain a state of pluripotency or even totipotency that is normally reserved only for embryonic or early larval stages. This reacquisition of stem cell-like functions is not a passive decay of cellular identity, but a highly regulated and active genetic campaign where the cell systematically clears its somatic memory and reactivates ancient signaling pathways that were once thought to be permanently deactivated after the initial formation of the Medusa. The key to this cellular magic lies in the interstitial cells, or I-cells, which are a population of multipotent stem cells found within the hydrozoan body that retain the ability to differentiate into various cell types throughout the animal's life. But in the specific case of the immortal jellyfish during its ontogenic reversal, even the fully specialized somatic cells appear to collaborate with or transform into these versatile I-cells through the process of transdifferentiation. This fluidity allows the organism to treat its own specialized tissues as a vast reservoir of raw biological material, capable of being melted down and recast into the structures required for a new juvenile life. As the jellyfish senses the environmental triggers for reversal, a wave of molecular signaling cascades through its transparent tissues, initiating a massive remodeling of the chromatin structure within the cell nuclei. This chromatin remodeling allows the transcriptional machinery of the cell to access genes that were previously hidden and inaccessible. Specifically, those genes involved in the formation of the colonial polyp and the initial budding of the stolon network. Simultaneously, the genes responsible for maintaining the adult Medusa's complex muscular and nervous systems are systematically downregulated, leading to the characteristic shrinkage and reabsorption of the umbrella and tentacles. This cellular plasticity ensures that no part of the organism's biological investment is wasted, as the energy and material from the aging Medusa are directly repurposed to fuel the rapid growth of the new polyp colony. The scientific study of this plasticity has profound implications for our understanding of regenerative medicine, as it demonstrates that the state of differentiation is not a one-way street, but a fluid and navigable landscape. If the molecular cues and genetic switches that control this plasticity in Turritopsis Dohrnii can be fully decoded, it may provide a roadmap for inducing similar levels of repair and renewal in the tissues of other species, including humans, where the loss of cellular plasticity is a fundamental driver of age-related decline and the inability to regenerate vital organs. Within the quiet, dark silence of the ocean depths, this microscopic dance of cellular identity continues to defy the laws of biological finality as the jellyfish's cells move seamlessly between specialized function and embryonic potential, proving that the boundary between a mature adult and a developing juvenile is far more porous than previously imagined. This endless cycle of shedding old identities to embrace the potential of the new is the essence of the jellyfish's resilience, allowing it to persist through time by constantly returning to the cellular starting line. As the organism settles onto the seabed in its cyst-like form, its cells are already busy weaving the future of the new colony, guided by the same ancient genetic blueprint that has allowed this species to navigate the global oceans for millions of years without ever reaching a final biological end. Each cellular transformation is a silent testament to the incredible versatility of life, and the reminder that even in the most specialized forms, the seed of a new beginning remains hidden, waiting for the right signal to bloom once again into the fullness of a renewed existence. A fundamental prerequisite for the biological immortality observed in Turritopsis Dohrnii is an unprecedented level of genomic stability. which the organism must diligently maintain across its countless life cycles and repeated ontogeny reversals. For almost every other multicellular species, the aging process is characterized by a gradual and cumulative accumulation of oxidative cellular damage, double-strand DNA breaks, and the progressive silencing of repair genes, leading to the terminal state of cellular senescence and eventual mortality. Yet, the immortal jellyfish possesses a robust and highly active suite of specialized DNA repair mechanisms that appear to operate with near-perfect fidelity, a phenomenon that is central to its ability to reset its biological clock with such precision. At the heart of this stability is a sophisticated regulatory system that ensures the constant vigilance of its genetic code, constantly monitoring for and correcting the myriad of DNA lesions caused by oxidative stress, environmental toxins, ultraviolet radiation, and the intrinsic errors that occur during the massive cellular divisions required to rebuild its body plan. Turritopsis Dohrnii likely utilizes superior base excision repair (BER) and nucleotide excision repair (NER) pathways, allowing it to efficiently identify and remove damaged bases and bulky adducts that would otherwise corrupt its genetic instructions. Furthermore, its cells must possess a highly effective system for repairing the most lethal form of DNA damage, the double-strand break (DSB), which would otherwise lead to catastrophic chromosomal rearrangements and cell death. This repair may involve both non-homologous end joining (NHEJ) and homologous recombination (HRR), which uses the identical sister chromatid as a template to rebuild the broken strand with extreme accuracy, ensuring the perfect reconstruction of its genome during the rapid tissue transformations of transdifferentiation. A crucial and unique component of this genetic preservation is the maintenance of telomeres, the repetitive DNA sequences that cap the ends of all linear chromosomes and protect them from degradation. For most organisms, telomeres shorten with each cell division, eventually triggering a permanent growth arrest known as the Hayflick limit. This cellular lifespan is thought to be a key driver of organismal aging, but in the specific case of the immortal jellyfish, it appears that the enzyme telomerase, responsible for extending telomeres, may be exceptionally active and constantly functional during the ontogeny reversal. This allows the cells to regain an embryonic-like lifespan and essentially reject the Hayflick limit, allowing the organism to restart its life cycle indefinitely. This relentless focus on genomic maintenance suggests a dynamic and ongoing rejection of the disposable soma theory, which posits that organisms must strategically allocate their limited energy resources between reproduction and repair, resulting in an inevitable decline. Turritopsis Dohrnii has effectively broken this evolutionary trade-off, demonstrating to the scientific world an unparalleled investment in somatic preservation and repair efficiency. As the organism collapses from a free-swimming Medusa into a cyst-like state, its genetic machinery is not shutting down, but rather shifting into a hypervigilant mode of surveillance, working to purge any mutated cells and correct any accumulated damage. This systematic cleansing ensures that when the new polyp colony emerges, it is built upon a flawlessly maintained genetic blueprint, which is free from the molecular scars of time. Within the quiet, cold silence of the ocean depths, this microscopic and perpetual work of genetic repair continues, serving as the silent foundation of the jellyfish's immortality and highlighting the incredible resilience of life when it is dedicated to maintaining the perfect information of its own unique blueprint, which can be found in the microscopic architecture of its DNA, making this creature a living museum of pristine genomic data. The intricate biological regulation of apoptosis, or programmed cell death, constitutes one of the most fundamental checkpoints in the life cycle of every multicellular organism inhabiting the terrestrial and aquatic realms, serving as a vital homeostatic mechanism to remove aged, damaged, or potentially deleterious cells from the systemic whole. in most complex life forms. This process is governed by a highly conserved and irreversible cascade of molecular events known as the caspase-dependent pathway, where specific proteolytic enzymes act as executioners to systematically dismantle the cellular architecture, resulting in the quiet and efficient removal of the cell without triggering an inflammatory response in the surrounding tissues. However, in the extraordinary biological narrative of Turritopsis Dohrnii, the standard logic of apoptotic finality is systematically interrupted and suppressed during the critical phases of ontogenic reversal, allowing the organism to preserve its cellular material even when faced with conditions that would normally trigger a terminal death signal. This selective inhibition of apoptosis is a cornerstone of the jellyfish's ability to transition from a sexually mature Medusa back to a juvenile polyp, as it prevents the mass loss of specialized tissues that must instead be repurposed through the process of transdifferentiation at the molecular level. This resistance to programmed death appears to involve a sophisticated upregulation of anti-apoptotic proteins, likely belonging to the widely studied BSCL2 family, which act as vigilant guardians of the mitochondrial membrane. By maintaining the structural integrity of the mitochondrial mitochondria, these proteins prevent the release of cytochrome C into the cytoplasm, thereby holding the formation of the apoptosis and the subsequent activation of the executioner caspases. This molecular shield allows the jellyfish to navigate the high-stress environment of its morphological collapse, turning what should be a terminal event into a period of deep cellular reorganization. Furthermore, the suppression of cell death in the immortal jellyfish is not merely a global shutdown of the system, but a highly localized and strategically managed phenomenon, where the organism distinguishes between cells that are beyond repair and those that possess the plasticity required for the next life stage. This level of cytokinic control suggests that Turritopsis Dohrnii possesses an advanced internal surveillance system that can modulate death signals in real time, ensuring that the energetic investment represented by its specialized cells is not wasted through premature degradation. As the Medusa bell begins to contract and the tentacles are reabsorbed into the central mass, the anti-apoptotic machinery works tirelessly to keep the internal cellular environment stable, providing the necessary time for the genetic reprogramming and chromatin remodeling to take effect. This bypass of the biological mandate for death is what allows the jellyfish to essentially bridge the gap between two entirely different body plans, maintaining a continuous thread of life across a chasm that would be fatal for any other creature. The scientific study of this apoptosis inhibition offers profound insights into the nature of cancer and degenerative diseases in other species, where the regulation of cell death has either become hyperactive or dangerously suppressed. By observing how the immortal jellyfish masters this balance, researchers hope to uncover the specific molecular switches that could one day be used to enhance tissue survival in human medicine, or to trigger the removal of senescent cells that drive the aging process in the silent and lightless depths of the world's oceans. This microscopic battle against the finality of death continues without pause, as the jellyfish's cells remain suspended in a state of perpetual potential, refusing to succumb to the standard entropic pressures that govern the rest of the biological world. The calm and rhythmic pulses of the ocean's currents mirror the quiet persistence of these cells, which carry within them the ancient and hard-coded instructions to persist, to adapt, and to begin again. As each potential death signal is intercepted and neutralized, the organism reaffirms its status as a biological anomaly, a creature that has found a way to exist outside the traditional boundaries of time and mortality, proving that even the most fundamental biological laws can be rewritten in the pursuit of eternal life. This mastery over the molecular signals of the end is what truly defines the spirit of the immortal jellyfish, turning every ending into a silent, dark, and peaceful beginning. The profound transformation from a free-swimming Medusa to a sessile, anchored polyp colony is not merely a structural reorganization, but a complex and highly specialized metabolic negotiation, one that demands extraordinary adaptation from the organism's internal energetic systems, and this level of adaptation is crucial for the survival of the species during the critical phase of ontogeny reversal. As the Medusa form senses the onset of environmental stressors that trigger this reversal, such as starvation, a drastic temperature drop, or physical trauma, it initiates a pre-programmed sequence of energy-conserving adaptations that override its standard physiological functions. The active, power-hungry swimming muscles of the umbrella, which in the Medusa phase consume a disproportionate amount of the total caloric intake to produce the rhythmic kinetic motion necessary for locomotion and feeding, are systematically deactivated, resulting in a swift and drastic reduction in overall mechanical energy expenditure. Simultaneously, the long, fragile tentacles, with their resource-intensive stinging cells, are reabsorbed into the body mass. Their complex protein structures and specialized lipids are broken down through intracellular digestion into their constitutive amino acids and simple sugars, which will serve as the essential fuel source for the ensuing transdifferentiation process. This process signifies a fundamental shift from a heterotrophic, active, and external resource-dependent existence to an internal, cannibalistic, and localized form of resource utilization. During this transitional phase, which can be thought of as a period of biological diapause, the overall metabolic rate plummets, a phenomenon that can be quantitatively measured by a significant reduction in oxygen consumption, reaching levels characteristic of a dormant or quiescent state. At the molecular level, this massive metabolic shift is precisely regulated by complex genetic and epigenetic control mechanisms, where the expression of specific mitochondrial genes is reconfigured. The cell begins to shift away from anaerobic glycolysis towards a more efficient, aerobic form of oxidative phosphorylation, ensuring that the available internal resources are maximized for the production of adenosine triphosphate, the essential energy currency for tissue remodeling and cellular reprogramming. Specific enzymatic pathways, like the pyruvate dehydrogenase complex and the various enzymes of the tricarboxylic acid cycle, are modulated to prioritize the synthesis of the molecules required for the conversion of specialized Medusa tissue into unspecialized polyp tissue. The level of control over metabolic pathways that Turritopsis Dohrnii demonstrates during this process is remarkable, as it allows the jellyfish to bypass the energetic limitations that would typically prevent such profound morphological transformations in other organisms. The organism effectively reallocates its entire energetic portfolio, shifting focus from external resource acquisition to internal structural reinvestment, prioritizing survival over procreation, locomotion, and even active defense, ensuring that when the new polyp colony emerges, it does so with a full complement of functional cells and vital energy reserves, which were meticulously recycled from its former adult self. This ability to navigate extreme resource constraints and adapt its metabolism in real time is an essential component of its resilience, allowing it to cheat death by transforming a terminal state into a period of energetic optimization. Within the quiet, dark silence of the ocean depths, this complex, microscopic, and perpetual process of metabolic optimization continues, a silent and unseen testament to the jellyfish's masterful adaptation to its environment and its unwavering persistence through time. Serving as a model for understanding extreme metabolic resilience and the unique potential for biological systems to reprogram their energetic demands, as the organism settles into its dormant, cyst-like form on the seabed, its cells are already busy weaving the future of the new colony, guided by the same ancient genetic blueprint that has allowed this species to navigate the global oceans for millions of years, utilizing every available molecular resource to ensure a new beginning. The remarkable transformation from a free-swimming Medusa to a sessile polyp colony is not merely a structural reorganization, but a complex and highly specialized response to external environmental cues. one that demands extraordinary adaptation from the organism's internal genetic systems. And this precise environmental connection is crucial for the survival of the species during the critical phase of ontogeny reversal, as the Medusa form senses the onset of environmental stressors that trigger this reversal, such as starvation, a drastic temperature drop, or physical trauma. It initiates a pre-programmed sequence of adaptations that override its standard physiological functions. In the standard biological model, an organism will typically respond to stress through traditional hormonal pathways or metabolic adjustments. However, in the case of the immortal jellyfish, the environmental stress signal is transmitted directly into the heart of the cellular nucleus, where it interacts with the sophisticated regulatory layer of information known as the epigenome. The epigenome acts as the master regulatory switchboard of the cell, deciding which genetic instructions are active and accessible, and which remain silenced and tightly packaged, without changing the underlying DNA sequence itself. Under stable, favorable conditions, the epigenome of the Turritopsis Dohrnii Medusa keeps the genetic programs associated with the free-swimming adult stage operationally active while firmly locking away the instructions required for the development of the polyp form. But when the organism is exposed to a life-threatening stress factor, it activates a set of ancient, highly conserved signaling cascades, leading to the rapid and widespread restructuring of its cellular identity. One of the primary mechanisms through which this environmental signal alters gene expression involves the complex and dynamic remodeling of chromatin structure. Chromatin, the material composed of DNA and histone proteins, can exist in either a tightly condensed, repressive state or a relaxed, open state that allows for gene transcription. Environmental stressors trigger specific epigenetic enzymes that modify the chemical tags on both the DNA and the histones, leading to profound shifts in chromatin accessibility. This process, which can be visualized through detailed models of DNA methylation and histone modification, effectively allows the jellyfish to open up the genetic blueprint for the polyp colony while silencing the code for the Medusa. Another key component of this stress response is the activation of specialized molecular chaperones known as heat-shock proteins, which are rapidly synthesized by the cell in response to conditions such as thermal stress or physical injury. These proteins play a critical role in stabilizing unfolding proteins and preventing cellular aggregation, ensuring that the transdifferentiation process can proceed in a highly organized manner. And specific models of the heat-shock protein mechanism help us understand how these molecules facilitate survival under extreme conditions. The sensitivity of Turritopsis Dohrnii's epigenome to its environment demonstrates an incredible level of evolutionary plasticity, allowing the species to bypass the standard rules of biological finality and trade the immediate energetic demands of adult life for a chance at a new beginning. The level of control that the jellyfish exercises over these epigenetic switches is remarkable, as it allows the organism to reverse its developmental fate in direct response to the same environmental challenges that would typically lead to death in other organisms, ensuring that every available molecular resource is strategically deployed to ensure its survival and its unwavering persistence through time, serving as a model for understanding extreme biological resilience and the unique potential for environmental cues to directly reshape cellular life as the organism transitions from Medusa to polyp, guided by the same ancient genetic blueprint that has allowed this species to navigate the global oceans for millions of years, utilizing every available signal to ensure its continuity. The recent advancements in high-throughput genomic sequencing technologies, including long-read sequencing platforms such as Pacific Biosciences and Oxford Nanopore, alongside short-read Illumina sequencing and high-C scaffolding for chromosome-level assembly, have provided the scientific community with an unprecedented window into the complex molecular architecture of the species Turritopsis Dohrnii. allowing for a comprehensive and high-resolution mapping of the specific gene groups that facilitate its unique and extraordinary ability to cheat the biological clock and navigate the waters of time by performing rigorous comparative genomic analyses between the immortal jellyfish and its close biological relative, Turritopsis rubra, which notably lacks the same robust and predictable capacity for ontogeny reversal. Researchers have been able to pinpoint the precise genetic deviations and expanded gene clusters that underpin the phenomenon of biological immortality. This extensive comparative work has revealed a significant and statistically relevant expansion in various gene families associated with the fundamental processes of DNA replication, DNA repair, and the long-term protection of telomeric sequences at the ends of chromosomes, suggesting that the jellyfish's evolutionary success is primarily built upon a superior and highly redundant system of genomic maintenance and surveillance. The genome of Turritopsis dohrnii is notably characterized by an increased copy number of genes involved in the systemic response to oxidative stress, such as superoxide dismutase and catalase homologues, as well as those regulating the checkpoints of the cell cycle and the intricate pathways of apoptosis, which work in perfect harmony to prevent the gradual accumulation of somatic cellular damage that typically drives the irreversible aging process and physiological decline in almost every other known species on the planet. One of the most intriguing and revolutionary findings in these recent sequencing studies is the identification of specific genes involved in the regulation of cellular plasticity and the process of transdifferentiation, such as the GLI-like genes, members of the SOX family, and various homobox gene clusters like the HOX and para-HOX genes, which act as the master architectural regulators of developmental timing and tissue identity during the organism's transformation. These genes appear to be highly conserved across the animal kingdom, yet uniquely and precisely regulated in the immortal jellyfish, allowing for the seamless and efficient transition between the complex, free-swimming Medusa form and the simple, juvenile polyp stage. Furthermore, the high-resolution sequencing data strongly suggests that Turritopsis dohrnii has evolved highly specialized and efficient mechanisms to maintain the stability and integrity of its mitochondrial genome, ensuring that the vital energy production factories within each cell remain fully functional and efficient across multiple life cycles and repeated morphological resets. The analysis of the jellyfish's transcriptome, which represents the complete set of RNA transcripts produced by the genome at any given time, has also provided critical and dynamic insights into how these genes are expressed in direct response to environmental triggers like starvation or physical trauma, revealing a highly coordinated and sequential wave of genetic activity that precisely precedes the physical reversal and structural collapse of the organism. The scientific study of these specific gene groups is not merely an academic exercise in marine biology or invertebrate zoology, but represents a vital and necessary step toward understanding the universal and fundamental principles of aging, regeneration, and cellular life. As the genomic data continues to be refined and the functional roles of these genes are validated through advanced techniques, it offers a powerful potential roadmap for identifying analogous or similar pathways in higher organisms, including humans, where the targeted manipulation of these ancient and dormant genetic switches could one day lead to significant breakthroughs in the field of regenerative medicine and the treatment of currently incurable age-related diseases and degenerative conditions. The vast, complex, and largely uncharted landscape of the jellyfish's DNA, hidden within its microscopic, fragile, and transparent body, serves as a profound living library of evolutionary survival strategies, refined over hundreds of millions of years of existence in the deep and silent currents of the global oceans. And as we continue to decode and interpret this complex genetic language, we find ourselves closer than ever to understanding the true and fluid nature of biological time and the infinite potential for life to persistently renew and sustain itself against the forces of entropy. This relentless pursuit of genetic and molecular knowledge represents a critical bridge between the ancient and forgotten mysteries of the sea and the future of human medical science. As the secrets of the immortal jellyfish are slowly but surely revealed through the power and precision of modern genomic tools, providing a glimpse into a world where aging is not an end, but a challenge to be overcome through the mastery of the information contained within the double helix. Every sequence and every gene analyzed brings us a step further in this silent journey. As the jellyfish continues its eternal dance through the dark blue void, carrying with it the blueprint for a life that refuses to fade, the profound transformation from a free-swimming Medusa to a sessile, anchored polyp colony is not merely a structural reorganization, but a complex and highly specialized biological negotiation, one that demands extraordinary adaptation from the organism's internal structural environment, and this precise environmental connection is crucial for the survival of the species during the critical phase of ontogeny reversal, the extracellular matrix, ECM, which provides the physical scaffold and biochemical signals that define the complex morphology of the jellyfish, must be entirely dismantled and rebuilt to accommodate the radically different body plan of the polyp. For most complex multicellular organisms, this level of structural fluidity and controlled architectural collapse is simply impossible, as the tissues become locked into specific developmental fates enforced by rigid ECM components. However, in the case of the immortal jellyfish, the entire body plan must be treated as a temporary and navigable landscape, guided by ancient genetic instructions. When the Medusa form senses the onset of environmental stressors that trigger this reversal, such as physical trauma, it initiates a pre-programmed sequence of adaptations that override its standard physiological functions. The highly structured and mineralized matrix components that once provided structural support for the pulsating bell, including complex collagen fibers and specialized non-fibrillar proteins, are systematically digested and reabsorbed by a specialized class of remodeling enzymes such as the matrix metalloproteinases, which act as a biochemical cleaning crew to dismantle the jellyfish's former architectural integrity. Simultaneously, the organism begins to synthesize new ECM components, specifically designed for the needs of the emerging polyp colony, including new types of structural collagens, laminins, and fibronectin, which provide the essential framework for the proliferating and transdifferentiating cells to migrate, adhere, and organize into the anchored colony form. This process of controlled structural dissolution and dynamic rescaffolding demonstrates that regeneration in Turritopsis Dohrnii is not just about the cells themselves, but about the fluid interplay between the cellular and extracellular environments, ensuring the perfect transformation of genetic reprogramming into physical form. The organism reallocates its entire energetic portfolio, prioritizing survival over procreation, locomotion, and active defense, ensuring that when the new polyp colony emerges, it does so with a perfectly customized structural environment, which was meticulously recycled from its former adult self. This ability to rebuild its own skeleton and structural blueprint in real time is a central component of its resilience, allowing it to cheat death by transforming a terminal architectural collapse into a period of deep structural renewal. Within the quiet, dark silence of the ocean depths, this complex, microscopic, and perpetual process of structural renewal continues. A silent and unseen testament to the jellyfish's masterful adaptation to its environment and its unwavering persistence through time, serving as a model for understanding extreme biological resilience and the unique potential for biological systems to reprogram their very structure. As the organism settles into its dormant, cyst-like form on the seabed, its cells are already busy weaving the future of the new colony, guided by the same ancient genetic blueprint that has allowed this species to navigate the global oceans for millions of years, utilizing every available molecular resource to ensure a new beginning. The study of Turritopsis Dohrnii within the specialized field of Biogerontology, the scientific discipline dedicated to understanding the biological processes of aging, has fundamentally shifted the paradigm for how researchers approach the concepts of lifespan, senescence, and regeneration for decades. Aging was widely considered a unidirectional and inescapable biological force, characterized by the progressive accumulation of molecular damage, a decline in physiological function, and an increased probability of mortality. Traditional model organisms used in biogerontology, such as the nematode Caenorhabditis elegans, the fruit fly Drosophila melanogaster, and the mouse musculus, have provided critical insights into the conserved genetic pathways and environmental interventions, such as dietary restriction or genetic manipulation, that can modulate the rate of aging and extend lifespan. But they all share the common feature of inevitable unidirectional senescence. Turritopsis Dohrnii, however, stands alone as a unique model of true negligible senescence and developmental plasticity, offering a radically different biological template where aging is not a fatal disease but a temporary and reversible state. The focus of scientific inquiry into this Hydromedusa is not on how it can slow down the aging clock, but on the precise molecular and cellular mechanisms it employs to reverse it. This distinction is critical, as it provides a potential blueprint for not just mitigating age-related decline, but for restoring juvenile function, a goal that is at the heart of regenerative medicine and anti-aging research. By analyzing the jellyfish's genome, transcriptome, and proteome during its ontogenic reversal, biogerontologists have identified specific clusters of genes that appear to be upregulated or highly active during this period. These include genes associated with enhanced DNA repair fidelity, allowing the organism to maintain perfect genomic information even after multiple life cycles, and genes for telomerase regulation, which extend the protective caps at the ends of chromosomes to prevent the cellular state known as the Hayflick limit, a fundamental driver of aging in other species. Furthermore, T. Dohrnii exhibits superior systems for maintaining proteostasis, the quality control and balance of all the proteins within the cell, preventing the aggregation of misfolded or damaged proteins that are a hallmark of human neurodegenerative diseases like Alzheimer's and Parkinson's. Its cells also seem to utilize alternative metabolic pathways during the reversal, maximizing energy efficiency and minimizing the production of oxidative reactive oxygen species, which contribute to tissue degeneration in aging humans. The implications of this research extend far beyond marine biology, as many of these key genetic pathways are ancient and highly conserved across the animal kingdom, meaning they are also present, but typically silenced or less effective. In human cells, the understanding of how T. Dohrnii manages to reactivate these pathways holds the potential to one day lead to therapeutic interventions for a vast array of human age-related diseases, neurodegeneration, cardiovascular failure, and the general decline in tissue regeneration capacity. It provides clues on how to induce cellular plasticity in human tissues with low regenerative potential, such as the heart and the spinal cord, where stem cell exhaustion is a major limitation, while other model organisms have shown that lifespan extension is a complex and polygenic trait. T. Dohrnii demonstrates that it is physically possible, at least at the hydrozoan level, to entirely circumvent the standard biological lifespan mandate, making it a critical and irreplaceable model for understanding extreme biological resilience and the unique potential for biological systems to reprogram their energetic demands and reset their biological clock in the silent, dark silence of the ocean depths. This complex, microscopic, and perpetual process of biological time negotiation continues, a silent and unseen testament to the jellyfish's masterful adaptation to its environment and its unwavering persistence through time, serving as a model for understanding not just aging, but the persistently and perhaps infinite potential for life to persistently renew and sustain itself against the force of entropy. Every scientific observation, every successful genomic sequence, every discovery of a new regulatory RNA or specialized protein brings biogerontology a step closer to deciphering the ultimate rules of existence, and T. Dohrnii remains a beacon of biological mystery, a small, transparent, and profound reminder that the limits we currently perceive as immutable may in fact be barriers waiting to be understood and eventually dissolved by the unrelenting curiosity of the human mind, seeking to understand the deep nature of biological time. The final and most comprehensive analysis of Turritopsis dohrnii within the context of the vast, complex, and competitive marine ecosystem focuses on deciphering the ultimate evolutionary advantages and inherent limitations of its unique capacity for biological immortality. For hundreds of millions of years, this small translucent hydrozoan has navigated the global oceans, utilizing every available environmental cue to ensure its genetic continuity, and its ability to systematically reset its biological clock stands as a masterful and unparalleled adaptation to its environment, providing a profound form of evolutionary resilience. In the standard biological paradigm, species must immense energetic resources into either somatic maintenance for individual longevity or into rapid reproduction for species survival, a critical trade-off that typically forces a unidirectional journey towards senescence and death. But T. dohrnii has evolved a molecular mechanism that strategically navigates this chasm, effectively prioritizing survival over immediate procreation through its unique ontogeny reversal. This provides the species with a critical advantage during periods of extreme environmental stress, such as prolonged starvation, physical trauma from predation, or catastrophic shifts in water temperature, conditions that would be terminal for almost all other pelagic organisms. By reabsorbing its specialized tissues and repurposing its cellular material to rebuild a youthful polyp colony, the jellyfish avoids the high metabolic cost of searching for new resources or partners, essentially recycling itself to ensure a new beginning. Every successful reversal is a victory of natural selection, allowing the exact same genetic individual to persist across multiple generations and deep geological time, maintaining perfect genomic information through a continuous process of purification and repair. However, this biological immortality is not synonymous with invincibility, and it carries with it significant evolutionary limitations. The individual Medusa remains highly susceptible to predation and can be consumed by a wide array of larger marine organisms, from other cnidarians to diverse fish species. Furthermore, it is not immune to the devastating effects of parasitic infections or diseases, which can lead to rapid systemic failure before the reversal sequence can be initiated, and the immense energetic cost of the transdifferentiation process to completely melt down specialized tissues and recast them into a juvenile form places a substantial metabolic burden on the organism, limiting its overall reproductive output and its capacity to rapidly colonize new territories. Every cycle of reversal and renewal represents a profound energetic investment, a strategic redirection of resources that prioritizes persistence over expansion. The environmental triggers that activate the reversal sequence are themselves indicators of extreme stress, suggesting that this adaptation is an evolutionary strategy of last resort, rather than a sustainable long-term mode of existence. The jellyfish's persistence is a delicate and dynamic balance between the potential for eternal life and the harsh, unrelenting realities of oceanic survival. It is not the largest or the strongest organism that necessarily survives, but the one that can most efficiently navigate the flow of time, utilizing every available molecular and genetic resource to ensure its unwavering persistence through the vast, cold currents of the global oceans. The unique potential for biological systems to reprogram their energetic demands and reset their biological clock, as exemplified by this species, remains a beacon of evolutionary ingenuity, a small, transparent, and profound reminder that the limits of life are far more fluid and non-linear than previously imagined. Every scientific observation, every successful genomic sequence, Every discovery of a new regulatory RNA or specialized protein brings us a step closer to understanding the true and fluid nature of biological time and the persistent, perhaps infinite, potential for life to persistently renew and sustain itself against the relentless forces of entropy. And as we continue to peer into its transparent structure, we find ourselves looking into a mirror that reflects our own quest for longevity and the eternal mystery of the cycle of existence.